Okay. All right, can you see the slide? Okay.
Yeah.
So I wanted to bring us back. I know we just had the midterm, and it's the last thing you want is to have to start thinking in the future long ways away. But we have our outline that is due on April 5th for the midterm, for the paper. Okay, so if you remember way back to the first day, we talked about how you'll have a term paper that's due at the end of the semester, but you have to turn in an outline and we go over it together and you give it to a peer review partner as well. And that is due on the 7th, on the 5th, and then we will schedule 2 days the next week on either the 7th or the 12th, where half of you will meet with me on one day and the other half on the other day. And so what that means is you need to be thinking about your term paper because as it says in the syllabus, you need to submit to me 2 weeks in advance your term paper idea, your topic. Okay. So over the next couple of weeks, you need to come up with some topics. I typically recommend you send me your top 3 topics that you'd like. Um, and so take a look. I believe I've posted the assignment and rubric document for the term paper, but if I haven't, I will do it later today. And so what you need to do is be thinking about, think about some of the topics we've covered thus far, or go ahead and look ahead in the textbook or look at the syllabus and see if there's anything you might be particularly interested in covering for your 10-page paper. And you're going to email me by March 22nd with your topics. It's first come, first serve. So if 3 people send me an email saying, I want to do it on hydrothermal vent diversity, the first one who sends me the email will get that topic. Okay. So please do start thinking about this. And then once we get our list and we get things sorted, I'll assign the peer review partnerships as we go forward. Okay. So I just have some tips to kind of get your mind frame going on this. So you will need to find the references and put them into your outline. Okay. So you don't have to have every single one of them, but as it says in the handout, I think I want about half of them in your outline. I will then suggest, oh, you should cut this, add this, et cetera, et cetera. So it doesn't have to be your final bibliography, so to speak. Most of these papers should be recent, but particularly if your topic is something that has been studied for many years, you can come up with a few of those, what we call seminal papers, early crucial papers from that field. Okay. This is not the same as our paper summaries. Okay. You're going to be integrating and taking snippets from all of these papers together and weaving them into a single cohesive story. So it's not exactly the same as what we've done for the summaries. And it's not a lab report, right? So you need to be thinking of this more like a review paper, right? So hopefully you guys have had a chance to read a review paper at some point. I know we haven't thus far in this class, although actually I take that back because the Tyson and Banfield paper was a review paper, right? So typically it's going to have an intro and then it's going to have sections, usually with headings that cover, 2 or 3 major points or topics. And so the literature review you do will provide the information that you'll fill in for those topics. So for example, a topic might be, what are the 4 ecosystems where viruses have been studied? And then you would expect in that section that you would be talking about papers that have addressed virus ecology in those 4 different habitats. And then at the end, you're going to have a conclusion, which should be synthesizing the topics you've reviewed in the term paper, as well as kind of coming up with future studies that might be done and the significance of it. Okay. So this is the just sort of outline of what I'm going to be looking for. And so think about topics. If you send me the email, I'll tell you, oh, that topic is too narrow, you won't find enough on it, or maybe it's too broad and I want you to refine it and narrow it down a little bit. So I'll give you feedback on those in addition to just saying, okay, yay or nay, you can have that topic or not. Okay. So I see somebody posted something in the chat. Let's check it out. So you email us by March 12th. You can start later today if you want, but the topics are things, but everybody needs to email me by March 12th. Okay, with 3 topics, 3 potential topics. If you just send me one and it's already been taken, then we have to go back and forth and back and forth. So try to think of 3 paper topics you'd be interested in. Every once in a while, I tell a student that none of those would be suitable or they're not available, and then we may have to go back and forth a little bit. But usually everybody gets something in their top 3. All right, any other questions? All right. So the good news is this outline is worth a lot of points in and of itself. And so, you know, even if you don't do very well on this first midterm, we have a lot of points left this semester through these other types of assignments. And so if you're better at writing papers than you are at taking midterms, especially in a COVID kind of environment, you know, we'll get through this, shall we say. All right, and if you can't find references to put in your bibliography, one good trick is to find like a key paper, like a review paper on your topic, maybe from the last 5 or 10 years. And if you find that paper, then you could look at their bibliography and find additional papers that they summarized. In fact, if there's interesting papers cited in a review, you should always go back and read the original article. You should never take a review article's word for it on what those other papers mean or how to interpret them. You should always go back and read the earlier literature. Okay. All right. So as I said, if I haven't already, I think I did, but if I haven't already, I will be posting the rubric and the assignment for the term paper later today. All right, so today and Wednesday, our lecture is on microbial population ecology. So population ecology is a very broad field of study, particularly in the sort of macroecology realm. And there's lots of terminology, and I think you've probably heard quite a few of these. In fact, we've talked about some of them in class today, or to date, I should say. Okay, so what I'd like you to do is we're going to go into breakouts, and I'd like you to define each of these, like come up with a consensus definition amongst yourselves on what you think a habitat is, a species is, a niche is, or niche, or a population, or a competition. So what do those terms mean to you? And I'll post these 5 in the chat as well so you see them. Okay, and you should have them on your slides now that you can access them. So let's go ahead and create the breakout rooms. We're going to do 5. And all right, so go ahead. Do you see the invitation? Carlos, can you see the invitation to go to the breakout room? And Catherine? All right. So does somebody want to share maybe their definition that the group came up with for habitat?
Yeah, I could go. What we came up with was so a certain set of abiotic and biotic factors that allow for an organism to adequately, live in that place.
Okay, so I'm hearing a place, and then there's a bunch of factors that would contribute to whether something could live there or not. And we may revisit that when we talk about the ecological niche concept. So, okay, did anybody else have a different take on the habitat?
We said something along the same lines. We said like geological locations where a species grows optimally, like in the best shape.
Okay, although I don't know that that's always the case, right? I mean, you could have an organism like a fish out of water that isn't optimally adapted to a specific location, at least in a short term, right? If it's lived there for a long time, then presumably it's adapted. But so we do have to remember we talked about transience and movement and things like that. But so when I think of the habitat, I tend to think of it being more in terms of the physical location. Um, and then the organisms that live there may be a combination of residents as well as sort of short time transitors or things like that. All right, does another group want to talk about what they said for species? How do we define species? Let's see, somebody put something in the chat. Caden says species would be living organisms that have the ability to transfer genes with each other or to one another. Okay, so that captures the idea of sort of living organisms and then gene transfer, which if you kind of remember what, sorry, if you remember what we talked about or maybe learned about in your general biology courses, there's that reproduction piece that's often connected with it. Johan, you put in the chat that organisms that are able to produce viable offspring, right? And you can take it one step further and say offspring that can in turn produce offspring. Because remember, if you have certain hybrids, like a mule, it might be sterile. So that's an offspring, but it's sterile, and so it's a dead end from an evolutionary standpoint. Microbes, we don't typically worry about this. Now, gene transfer is definitely something we're going to talk about, but the key there is what don't microbes have, or what kind of reproduction don't they utilize, which bears on these kinds of things that are being brought up.
Sexual.
Right, so they're asexual. So the reproductive piece, it's basically if you can grow enough to divide, that's all you need, right? Okay. So anybody else have a different thing or thought to add to the species discussion? If we're talking about asexuals and we're not really requiring them to interbreed, like you probably learned in general biology or in another class, what definitions should we be thinking about then? What are some ways we might think of defining species at the microbial level or the asexual prokaryotic level in particular? Any ideas? All right, well, we'll talk more in actually the next slide on this, so I'll save that. All right, so the ecological niche, what group had a thought on that? Anybody heard that term before? Have a thought as to how we might define that?
Yeah, group one, we pretty much said a specific environmental section colonized by a single species. due to metabolic needs.
Okay, so there's a sense of like the environment is creating or selecting for a species in this case. Could it be more than one species, do you think?
If I'm not mistaken, one niche is only populated by one species.
Okay, well, we'll talk about that in a little bit. So that's
Okay.
a good thing to think about. All right, that sounds good. Did any of the other groups? Let's see. It looks like a microenvironment of a certain population. So maybe part of the population is in this niche. Is that kind of what you're meaning, Johan? Okay.
Yes.
All right. So it may not be the entire global population of it, but in a specific location, maybe. So you can see how a lot of these terms interact with each other, right? Habitat can inform the niche. And of course, ultimately over time, that can then contribute to speciation events, et cetera. Okay. All right, is there anything else that any of the other groups wanted to contribute for the discussion of the ecological niche? All right, Angel says in the chat, a specific role an organism has in an environment. I like that definition because it kind of implies that they're doing something, right? They're active. But that role can also be informed by where it is and the interactions they might have. So, okay, that sounds good. And again, we'll come back to this as well. I just wanted us to have an initial discussion. Population. We've used this term before. In fact, we said with microbes, we are usually thinking about population. So what did your group think about populations, or how did you define it? Anybody?
Could it be the interactions between different organisms?
So the population itself is a group of organisms that would interact, but it's not really the interactions per se. Does that make sense? So Ashland says a group of one species in a particular location. Okay. Yeah, I like that because it's sort of the unit of measure. It may not be every organism in that species. It probably isn't. But being in a particular location gives it that sort of cohesiveness. And the other thing is that when we're talking about gene flow, we talk about gene flow a lot with populations with macro sort of organisms. But of course, with lateral gene transfer, that also would take place within a population and probably not between populations, at least in the short term. Okay. Anybody else have anything they wanted to contribute about population? Okay. Well, then our last term is competition. Who has a good definition for that? What does it mean when we say that organisms, individuals, populations? Marwa says an organism or a species fighting for resources in order to survive. Okay, so there's the struggle, right? Very Darwinian way of framing it, Marwa. But yeah, I like that. Anybody else? Everybody kind of agrees with that one. Okay. All right, well, let's go ahead. I do not have a slide really for defining habitat, but we're going to talk about speciations because I think habitat is fairly straightforward. And we had all of those lectures earlier this semester about habitats. So for species, we already kind of mentioned some of this, right? So the biological species concept that Ernst Mayr proposed decades ago was really based on animals and plants, right? Sexual reproduction. And so it really does not work for asexuals, especially like bacteria and archaea. There's another kind of concept for species, which is tied more to ecology. So we call it the ecological species concept. And so this would be the individuals that are identical in terms of their ecological interactions. And so we can actually use a term called an ecotype. And so this really combines the species concept and the niche-based concept in the sense that it's a specific genotype that lives in a specific habitat, for example, right? So this is really bringing all of these three terms together. And this is some work that was actually really pioneered by one of my former college professors, Fred Cohen. And so what he proposes is that ecotypes are really the dominant form of kind of speciation. And what happens is you'll have a mutation, a beneficial mutation, which allows one member of that population to be more fit or more, superior to the relatives that didn't mutate. And so periodically there will be natural purifying selection that will favor this newly arised genotype and will wipe out everybody else, typically by competition, right? So this new mutant that it becomes dominant and everybody else falls away. We've been hearing a lot about this with COVID, right? You guys have, I hope, are hearing about the UK variant and the South Africa variant, and there's a California variant out there, and all these different novel mutated types of the COVID vaccine, the COVID virus that seem to be replacing the original virus that we first identified in Wuhan, China. So these viruses are a perfect example of this ecotype. If they are spread or reproduced more rapidly, which seems to be the case, like with the UK variant, for example, then they will take over. Whereas the other original strains from which it arose may become much less prevalent in the environment. Does that make sense? So that's, essentially the COVID vaccine has an ecology and an evolutionary trajectory, much like what we're talking about in this class. But this would certainly apply to bacteria, archaea, microbial eukaryotes as well. All right. Now, another way of thinking about species is to use genetic definitions, because we can't ask the question, does this organism and that organism have sex? If so, we call it a species, right? In this case, we could sequence their genes or their genomes and ask the question, how similar, how related are they, right? Because of course, if you're one species, you should be very closely related at the genomic level. And in fact, even before genome sequencing was available, there's a technique called DNA hybridization, which was pioneered, where essentially you merge, you extract the DNA from a culture and another culture, and you see how much they stick together or hybridize after denaturing the double-stranded DNA. So if you think about a circular genome and another circular genome, if you unzip those and you ask the question, how much can they line up? If 70% of the total genome hybridizes or sticks together, implying that they're complementary, then we would call it one species. Now, with the advent of ribosomal RNA sequencing, a shortcut for that was developed. And so you'll see in many papers that a 97% cutoff or threshold is used. Some other authors have proposed that it should be more like 99%, but we're not going to get into that here. But if you took two 16S gene sequences and they were identical, then we would say for sure they're a species. But you can even be 99%, 98%, 97% identical, and we would lump them at least as a rough draft, right? You probably want to go in and do more thorough analysis of those groups, but we've used this and we actually have a definition then of something that we call an operational taxonomic unit, or an OTU. This is often used instead of species when we're talking about bacteria or archaea, because if we're going to use a genetic definition, then we're really not for sure whether it's a species or not, but we do know that it is a classification unit or a taxonomic unit. And so that's where this operational taxonomic unit definition comes from. Now, of course, we've talked about how horizontal transfer is really much more common than was originally recognized, say, 20 or 30 years ago. And we can think of this like bacterial or prokaryotic sex in a way, but of course it's very different because as we saw from the Chivian paper, you can actually get gene transfers that span entire domains, right? And what was the example that was given in the Chivian paper about that? Who remembers?
Nitrogen fixation genes.
Yeah, so some of the nitrogenase genes seem to be transferred from archaea to the desulfuridus, which was a bacterium, right? So all of this makes things very complicated. We all actually, including us, have archaeal and bacterial genomes, little snips of it in our genomes. So horizontal transfer seems to have crossed all three domains of life, but particularly are prevalent between bacteria and archaea. And incidentally, likely played a huge role in the origin of the earliest eukaryote, which may have involved a fusion of a bacterium and a eukaryote. The other thing it does is it makes it hard for us to categorize, or we have to be careful when we categorize if we're just sequencing part of the genome, because you can actually see horizontal transfer of genes. Now, one of the reasons we use ribosomal RNA is that it is less prone to horizontal transfer. However, it has been demonstrated that even 16S ribosomal RNA genes get transferred horizontally. And I hope you can appreciate that if that happens, that's going to lead to potential misclassification. Because if a cyanobacterial 16S gene gets moved over into a heterotroph, right, it's still going to be involved in translation. It's still going to be involved in all of the functions. This is not a photosynthesis gene. So it's actually quite possible to move something even from a phototroph to a heterotroph. But if you sequence the 16S gene from that heterotroph and it's similar to cyanobacteria, you could be totally wrong in what you think this organism represents in terms of their ecology, their metabolism, etc. Now, it's less common for that to happen, but it's highly common for metabolic genes to be moved. And these can be carried on plasmids or transferred via viruses, right? And we've already encountered that with the Chivian paper, right? So something like the nitrogenase gene would confer an important new function or maybe change the function of existing nitrogen fixation in the desulfuridus audaxpiator organism. Or it can bring a whole new function in, right? So maybe it was an organism that's not capable of using a certain substrate. And then the genes are brought in laterally, and suddenly that organism has a whole new lifestyle open to it. And if you appreciate the niche concept that we were just talking about a few minutes ago, this could radically change the species niche. So this is fascinating from one hand, but it's also complicating from another. So we do need to think of microbial genomes as being chimeric or mixtures, right? The chimera was the mythical creature that was the mixture of two different animals, right? Really, all microbial genomes are going to include genes that are ancestral to whatever that organism is, dating back millions to billions of years. But some of those genes also came from the outside along the way. And so this leads us to this interesting idea of trying to figure out what are the genes that define a species or a group of organisms. So, for example, there's been quite a lot of study of E. coli because we have thousands and thousands of E. coli genomes in the databases now. And so you could ask the question, what are the genes shared by all E. coli? And those shared genes would be something we would think about as the core genome. Essentially, what does it take to be an E. coli? Whereas there's going to be other genes that are found in some strains of E. coli, but not all, right? We know that there are pathogenic strains and non-pathogenic strains, right? But it turns out that there's large amounts of the genome, 20, 30, 40% of genomes within species that can be found in some, but not all members that have been cultured from a given species. And of course, if we have organisms only known from the environment, then we really have no way of even knowing. But this gives us this concept of, kind of this core genome versus accessory, right? And all of this can lead to kind of a blurring of those niche lines. And so this kind of comes back to what you were raising, Victor, in terms of you should only have one thing occupying a niche at any given time, but lateral transfer can make this complicated. So this is just a diagram that kind of shows this idea of, so there's a core genome that everybody has, but then there's this accessory genome that only some members have. And this may allow them to specialize, and it may also allow them to have a different niche so that they differentiate from their fellow species, fellow members of the same species. So some strains, or you could think of some populations will have, they all have the core, but then there's going to be different accessory genomes in different strains. Does that make sense? All right. Any questions about that? All right, well, let's see how much we are understanding from the definition I gave you of the operational taxonomic unit a few minutes ago. So what was the most common definition used to describe microbial species? Is it 97% whole genome hybridization, 70% 16S gene sequence identity, 97% 16S sequence identity, or 100% plasmid compatibility test results? So let me start the poll. Obviously, just pick A through D for this one and go ahead. So which of these is the most common OTU definition? Okay, go ahead and register your votes if you haven't already. All right. All right, so it looks like the vast majority of you put C, which is the correct answer. So good job. A couple of you put A. What is the difference between A and C? Why is A incorrect? Does somebody want to share that? Why would A be wrong?
If the whole genome was hybridized, wouldn't it be unfunctional? Does that make sense? Or am I looking at it wrong?
Well, the whole genome hybridization, we did talk about that is a test. Of course, it's not, these are not living organisms, right? To do these tests, you have to extract their DNA or their 16S genes and sequence them, right? So what we're really asking the question here is, how do the genomes compare? And in two of these answers, we're looking at just a single gene, the 16S gene. But in this first one, we're looking at the entire genome. And so if that's with that as our perspective, what would the percent be for the whole genome hybridization that we would be looking for? It's not 97% when you look at the entire genome. What is it?
70%.
It would be 70. And that makes sense, right? Because the whole genome is much more, right? So you would never expect that every gene across the entire genome, hundreds of genes potentially, would be so similar that it would be 97% similar. It's only those really slowly evolving genes like the ribosomal RNA that we said they don't change much, because if they do, they usually get removed from the gene pool because most mutations are negative, deleterious. So the bar is lower for whole genome hybridization, and it's only 70%. But in a sense, that should be roughly equivalent to 97% identity of a single conserved gene like the 16S. Does that make sense? So the best answer was C, as most of you put. But I just want to make sure everyone understands that. All right. Okay. So now we're going to start talking a little bit about some of the concepts that lead into competition. Okay. So the first thing is this graph. And what you want to look at here is, this is kind of a theoretical graph. What it's showing here is the utilization or usage of a given resource. Like, let's say it's food, right? Glucose, right? So how much glucose is used, and then on the x-axis here, it's sort of showing the variation in utilization of glucose of a population, right, across a resource gradient. So some members of a population will use glucose optimally at lower concentrations, maybe. Others may like it to be in really high concentration. And like many traits that are not determined by one gene. Remember, you probably learned in general biology about polygenic traits, or in many cases, populations represent a distribution of different phenotypes, right? Based on, kind of variation in the genotypes that underline that population. So some are better using high levels of it, some better using low levels, but on average, there's going to be a fairly normal distribution, so that most organisms tend to use in the middle, right? Okay, so a lot of traits are going to be normally distributed like this. And what you can see is that if you have a single species in isolation, then it can accommodate all of those different organisms, the high utilizers, the low utilizers, and everybody in between. Okay. But when we look at this second graph here, what is this showing, do you think?
Another species or population?
Right. So another species shows up, and these two species are now in competition with each other. And what you can see is that this other species might be geared so that the resource utilization is higher further up the gradient, right? So maybe this species B requires even larger amounts of glucose. That was our example, right? And so B may be evolved to utilize very large quantities of glucose, whereas they don't do very well, like down here, at very low levels of glucose. And so what you can see is that this too will have a normal distribution. And what this graph is showing is their individual, like ignore A, and this is what B would look like. Ignore B, and this is what A looks like, right? It's pretty similar to up here. But what we really care about here is this point of intersection where they overlap. And so where they overlap over time will tend to be differentiated, or what we call partitioned. So the B's will not do as well here. They're sloping down on their optimum. The A's also are kind of sloping down. And so what's going to happen is their curves here are a little bit narrower on this side. Do you notice that? And this side. Over time, what happens is the two species will diverge. And the reason for that is through differential survival and reproduction. If you think about it, the organisms in this middle part here are competing with themselves and with the other species. Whereas the members up here just compete with themselves, and the ones down here just compete with these guys. So the competition is much higher here. And so over time, they will tend to diverge in their utilization because of lower reproductive output of the populations here. Does that make sense? So fewer A's are going to survive and fewer B's are going to survive because of competition. And so over time, we'll tend to see that their utilization gradients reflect that. This actually opens up the case where a third species, C, could come in that is optimally attuned to the middle ground between them. Because at this point, we've actually removed a lot of the competition. And so C might come in and start competing. But then guess what's going to happen? The C's up here do worse. The B's down here do worse, and so on and so forth with A and C. And so over time, what happens is you are partitioning or divvying up the resource gradient across these three unique populations. Okay. And so this gives you an example of how phenotypic selection, right, leads to changes in resource utilization over time. And it's all due to these areas of overlap or competition. Okay. And so with each new species that comes in and arrives, it's leading to narrower resource utilization windows or distributions, right? So there's fewer high A's, fewer low B's, and the C's are kind of the Goldilocks. They're right in the middle, right? So both their high and lows are gone, and it's going to be the mean or the average C utilizers that will survive over the long term in the face of this competition. Does that make sense? All right. Any questions? So it's important for us to think about, this is just one resource we were talking about at a time, but of course that's not the way things go. And so really when we're thinking about the ecological niche, we need to think of this in terms of entire suites of conditions and resource utilizations that we need to worry about, which lead to survival and reproduction of a given species. Okay. So an ecological niche is really that whole range. You know, what is the temperature they can live under? What is the food source utilization? What is the space needs? How much oxygen? You name it, we could put it into one of these niche concepts. Pretty much anything that will define survival and reproduction can be mapped onto this. And we can do it one at a time. So we can think in terms of dimensions here, right? So maybe temperature is the first dimension. Maybe you have a strain of bacteria and they can live from, 15 degrees centigrade to 22 degrees centigrade. Okay, so that would be represented by this. But then they also have a humidity requirement, right? Or a moisture requirement in the air. So we need to map that. That would be a second dimension we'd be adding on. Okay. And then food, right? Food availability could be a third dimension, so on and so forth. So G. Evelyn Hutchinson in the 50s, so decades ago, came up with this definition, this ecological niche concept. And what they said was that it's an n-dimensional hypervolume. So if you map these, it's 3 dimensions, 4, 5, 6, 7, 8, all the way up to n dimensions, which is really like impossible to define, but that would really be the way we would have to wrap our head around all the possible abiotic interactions that can occur. What are those conditions under which the organisms can survive? Okay. Then we can overlay on that biotic interactions in terms of competition and things like that, which would further constrain or further narrow where these organisms are found, right? And that would take us from the fundamental niche, which is really this box is kind of the all possible conditions under which an organism can live. And that's what the fundamental niche is, right? Without needing to worry about interference from other species. But in reality, they're not going to be out there alone, right? They're going to be competing with other species in any environment that they're in. I guess the possible exception to this is our Desulfurudis, right? Because maybe they were on their own, mostly in that fissure. But in almost any habitat, you're going to be competing with other species for space, light, oxygen, glucose, you name it, right? And so the realized niche has to reflect the biological interactions, those competitive interactions or predatory interactions, anything that is going to reduce survival and reproduction. And so in every case, you'll end up with the realized niche being smaller than the fundamental niche, with the possible exception of symbiotic interactions, okay? But in general, what you're seeing here is that competition, predation, and these biological things, they constrain or they reduce where organisms can be found. Does that make sense? So there's the where could you live, but then where do you really live, right? You know, we could all live in Beverly Hills, but unless we're rich, we don't really live in Beverly Hills, right? So that's kind of what we're getting at. All right. So if we're going to think about this, we can take the concept I showed you earlier with the 3 different species and their resource utilization overlaps. And if we map it onto our sort of n-dimensional hypervolume, then you come up with these more complicated interactions, right? And so this kind of concept actually comes out when we look at the overlap of 3 species. And remember, this is 3 at a time, so it's simple, and it's only 2 dimensions at a time, so we can wrap our brains around this. But you can see that there's places where A is going to overlap with C, okay, and or with B, right? But the way they overlap could certainly be different in this dimension versus this dimension, right? And so it gets very complicated, okay? But this is kind of how you can then see where these organisms would end up being like sort of, it's almost like they're being boxed in, okay? All right. All right. So let's see if we have kind of followed this. So which of the following are important for determining the size of a population's niche? tolerance for abiotic conditions, competition, overlap in resource utilization, both A and B or E, all of the above. Go ahead and answer. Right, is that everybody? Oh, your answer. Okay. So it looks like with one exception, everybody really likes A and B as an answer. The question is C also the answer? And the answer is yes. E, all of the above is really the best answer. So we talked about initially about the tolerance for those abiotic conditions. We talked about competition, right? But really, competition is going to be intimately connected with this overlap in resource utilization. Essentially, what I'm talking about there is this, right? So these points of overlap define how A is distributed versus C versus B, right? Okay. So all of these factors will determine the size, and essentially they're going to shrink the size of a niche. Okay. Now I should highlight that these are theoretical constructs, right? We don't know every single aspect of every single species out there, right? It's unknowable. But this is an active area of research trying to understand, okay, well, this species lives here. Why does it live there, right? Right. These are the questions we can ask from an ecological perspective. And we can try to determine what those abiotic factors as well as competition and resource utilization might be. All right. So competition, we've used this word now repeatedly, but what we're really looking at here is, as a definition, is the interaction between populations. And the thing for us to think about is that growth is going to decrease for both. So even if you have one species is better at utilizing a resource than another, if you put them together, every, let's say unit of food, let's go back to our glucose. Every glucose species 1 eats is a glucose that species 2 can't eat, and vice versa. So you always have lower growth rates whenever you have 2 species competing with each other, even if one is a better competitor than the other. It's a lose-lose proposition. Okay. And competition can occur in multiple ways. It can occur, sort of directly. That's kind of often a physical thing. So there can be like interference. Maybe a fungal colony is living on a log and rotting it. If it got there first, then it's excluding another fungal colony from, some mold or mildew from coming over and occupying that space. That's interfering with its physical ability to be there. But in many cases, it's not so direct. It's often indirect. For example, if one species uses the carbon like glucose first, by definition, that carbon is no longer available for species 2. And so these indirect exploitation mediated effects are a little more indirect. Okay. All right. Any questions about those definitions? All right. When we talk about competition, we can actually model this with simple math. And you guys have all seen this before. We've shown it in this class, and you've all seen it in, those of you that are micro majors have seen it in general micro class. But you don't always know why that S curve exists. And this is really the kind of the mathematical definition of why we see lag, log, stationary phase, right? So if you take a strain of bacteria, let's say, I don't know, Klebsiella. You take a strain of bacteria and you put it in a flask with lots of medium and grow it overnight and come back the next day, right? You'll see a nice turbid growth in there. But if you measured absorbance every hour for 24 hours as that was happening, then what you would see is a typical growth curve, something like this, right? So what this models is the change in population, which we define as N. N is the number of individuals. In this case, it's the estimated cell count, right? The population size. It could be thousands, but it's oftentimes in a bacterial cell culture, it's often going to be millions to billions to trillions of cells that can grow in a flask, let's say, okay? And so what we can see is that there's that initial lag phase, and then there's that exponential growth phase that we've all experienced, all seen, right? But then eventually, what happens? The slope starts to decrease, and eventually we hit a limit for growth, okay? This limit is referred to as the carrying capacity, okay? What do you think is happening up here when you hit the limit? Who knows why this goes into stationary phase like this?
I think the number of cells dying and the number of cells increasing like reaches an equilibrium.
Okay. And why is that? Like, what causes the death rate essentially to go up?
Lack of resources.
A lack of resources, right? Intraspecific, meaning within the species. So in general, we think of, oh yeah, like, right, these are, let's say it's a bacterium like Klebsiella. So these are asexual clonal relatives, right? One cell becomes 2, 4, 16, you know, the whole 9 yards in terms of exponential growth. But eventually they use up something, right? And they don't have to use up everything in the growth medium. If one thing becomes limiting, game over, right? And that's what's reflected here with this kind of balance. But you're right, you're not in death phase. So it is at an equilibrium that there's as many are dying as are living and growing, or else they're just sitting there, right? They've stopped growing. They're not dying yet, but they're just kind of in stationary phase. Right? And this curve is actually modeled by this equation on the screen, which I know math, we as biologists sometimes get a little freaked out by, but you do need to understand this simple math, right? It's a standard differential equation. I know we probably blocked out differential equations as soon as we passed calc and we moved on, unless you're kind of a math type out there, which is great. But if you're not, don't worry, it's relatively simple. But what you need to do is understand that we essentially just have 3 variables here. As n goes up, you're going to see, you know, competition increasing, and you're going to be getting closer and closer to K, whatever that is, the carrying capacity. K could be 1,000 cells or 1 billion cells, right? It's not predefined. And the slope, right, is defined by R max, the intrinsic rate of growth. And the slope is going to be dictated by how fast a growing strain is. So some strains grow faster than others, and that's based on their metabolism and their biochemistry, and everyone is unique. And so that's reflected in this slope. So those are really the 3 things: the number of cells, how quickly they increase based on their R, and then where they level off in terms of their carrying capacity. So this is intraspecific competition. And ecologists looked at this and they realized that, you know, you have some organisms that are inclined to grow very quickly, and we call them R-selected. So these organisms are going to rapidly proliferate, whereas others don't grow as quickly, but they do better near the carrying capacity. When times are tough, competition is really high, and maybe they're better at competing. So we have these terms R-selected and K-selected. So R-selected species in general have very rapid growth rates, right? But they don't compete very well. Okay, so they like it down here when competition is low, you're not near the carrying capacity, and they can just grow as quickly as they possibly can. These are often good dispersers. And so this is a strategy that seems to have evolved in certain species that disperse, like maybe they form spores, they get into a location, they use resources, and they get out when competition starts to get fierce. By contrast, K-selected organisms are slow growers. They don't rush to do anything. So this is sort of the proverbial turtle and hare, right? So the K-selected are the tortoises. They just slowly go along, but they do very well when, you know, some of these R-selected species don't do very well. When there's lots of competition for resources, they're optimally attuned to be able to eke out a slow growth lifestyle under those conditions. Okay, and so we can sort of define species that way. All right. So competition, right? We said, is it good and good, or good and bad, or bad and bad for the two different species? What did we say about competition?
Bad and bad.
We said it's bad and bad, but it can be more bad for one versus another. And that's the case when you have competitive exclusion. If you have significant niche overlap, and this comes back to your point, Victor, from earlier, in general, we think of niches as being unique. A single species or ecotype is going to occupy that niche. And the reason is that if it overlaps significantly with another one, they're going to be competing, and typically one population will drive the other to extinction. This doesn't always happen 100% of the time, but it's very common. Okay. And in fact, if you do mathematical models, in some semesters, I usually show that, but this semester, I'm going to spare you guys that. You can actually see that in 4 different scenarios, 3 out of the 4 will drive to competitive exclusion or local extinction. Only in certain circumstances can you end up with competitive coexistence. And so this just kind of illustrates that, right? If you have the purple defining one species, the blue defining another, this is going to be majorly overlapping in that kind of sense we were talking about earlier with our resource utilization parameters. So either blue or purple is going to win in the long run. Okay. All right, I think I'm going to stop there because we've learned about the logistic equation in the earlier equation. Starting next time, what we're going to do is we're going to take that and model intraspecific competition, where we look at species 1 and species 2 and how they interact with each other. Okay. Any questions about what we talked about today? Competition, niches, any of that? All right. Well, we will pick this up next time right here, and I will see you guys on Wednesday. And as I said, I'll try to get those grades posted by class on Wednesday if possible.